For my final project, I'm going to be talking about the entourage effect. So let's jump right in. What is the entourage effect? The best way that I like to describe it is thinking about the whole being greater than the sum of the parts. So THC, delta 9, pictured here on the bottom right, we all know is pretty much the number one active ingredient of cannabis. But we also know that it doesn't act alone. The cannabis plant is known to produce thousands of phytochemicals, hundreds of cannabinoids, dozens of terpenoids, who knows how many flavonoids, lots of other compounds, and all of these in concert actually contribute to an effect greater than what you'd get from just delta 9 tetrahydrocannabinol in isolation. And we'll take a look at some of the scientific literature that supports this idea. So right off the bat, here is the paper from 1998 that first proposed the existence of the entourage effect. The last name in the authors list here hopefully looks familiar to some people. Dr. Raphael Mechoulam is really primarily responsible for a lot of what we know about cannabinoids. His lab was the first to synthesize THC, CBD, and some other cannabinoids, and really, again, are responsible for much of what we know about cannabinoids and the endocannabinoid system even. This paper is taking a look at one of the endocannabinoids that his lab discovered and isolated that exists naturally within the body and binds to the cannabinoid receptors within the body. They found that this molecule doesn't seem to act alone, that some other molecules that don't actually bind at CB1 or CB2 seem to mediate the effects of this endogenous cannabinoid. And this effect that they observed, they termed the entourage effect, and it's been pretty well studied in the literature for the last, just over 20 years since being first proposed. But let's back up just a touch and really quickly go over some basic background about cannabinoid receptors so that we can better understand what exactly I mean by the entourage effect as opposed to just the direct effects of THC binding to cannabinoid receptors. So remember, there are cannabinoid receptors, two different types, CB1 and CB2. CB1 primarily concentrated within the brain and central nervous system. CB2 found in a lot of different organs, skeletal muscle, bone, eye, all sorts of different places throughout the body. And THC, tetrahydrocannabinol, that main ingredient of cannabis, is the primary ligand that binds to cannabinoid receptors. So again, we can think about this like a lock and key. The cannabinoid receptor is a lock. The THC is the key that opens that lock. THC fits perfectly into that cannabinoid receptor in ways that other molecules just are not able to. And so it turns out that molecules that are not able to fit into that cannabinoid receptor and activate it directly might play a role nonetheless in the activation that occurs at that receptor. Once THC binds to that cannabinoid receptor, it then causes a cascading event to occur within this presynaptic cell, and it causes some neurotransmitters to be released. Those can have a huge variety of functions that are way outside the scope of this, but suffice it to say that THC is that lock, or rather the key, that fits into the lock of the cannabinoid receptor. And what many studies have now found is that molecules that don't bind directly to CB1 or CB2 are nonetheless able to impact the actions at those receptors. So this is another snippet from that paper by Ben Shabat et al. That first proposed the entourage effect, and they found that while the molecules they were looking at didn't directly bind to either cannabinoid receptor, they were able to potentiate, to affect and increase the apparent binding of an endogenous cannabinoid to both receptor types. And here we are, the same type of entourage effect was observed in several in vivo tests which are commonly used with cannabinoids. So all they're saying there is that they found that in real life tests, they observed this same effect. So moving on, here's a quick look at some of the data from that first paper that proposed the entourage effect. Without going too deeply into it, you can see in each of these four charts, the far right column, basically all we need to know is that that far right column, those asterisks on each of them, indicates that the results are significantly different from the others. And take a look at the key to this. The far right column in each of these four is that preparation that included the endogenous cannabinoid plus some other molecules, whereas the other columns are the endogenous cannabinoid and then the two additional molecules, the far left column being a control. And so again, the key takeaway here is that in four different measures of the effects of this endogenous cannabinoid, it was highest, the effect was the greatest when the endogenous cannabinoid was delivered in conjunction with a couple of molecules that did not have any direct activity at either CB1 or CB2. Here's the quintessential demonstration of what they mean by the entourage effect. Something that didn't bind directly actually had an effect on how strongly that binding occurred, or rather the effect of the binding ended up being. So in conclusion from that paper, they said that the effect of the active compound in presence of its entourage compounds may lead to observations of effects closer to those in nature than investigations with the active component only. So basically, using a whole plant preparation is going to have different effects than an isolate of THC or CBD alone. And we'll take a quick look at a few studies that came out since then in the intervening years that support this idea. Here's a neat one, actually. Overcoming the bell-shaped dose response of cannabidiol, CBD, by using cannabis extract enriched in cannabidiol. So basically what this study did is they took a look at the anti-inflammatory and anti-anxiety effects of CBD isolated and compared those with the effects of a botanical extract, meaning an extract containing all of the phytochemicals from the cannabis plant. And one of their conclusions here, the final thing I've highlighted on this slide, it is likely that other compounds in the extract synergize with CBD to achieve the desired anti-inflammatory action. And they say above that, makes this plant medicine ideal for clinical uses. So here's what they found. Take a look at the two left-hand charts. These are the effects of CBD as an isolate. Take a look. You can see there actually is a bell curve shape to the dose response. As you give more and more CBD, you don't actually see a higher and higher response, is the takeaway from these two left-hand charts. Take a look at the two charts on the right hand, though. The higher the dosage, the farther over to the right the columns are, the higher the peaks go. In other words, there's a linear response between the dosage given and the physiological response as a result when all the cannabinoids and phytochemicals involved are given together, that we do not see when CBD is given alone. This explains what was going on in that last slide in detail. We'll just skip right over that, but I wanted to put it in here in case anybody wants to read it. Here's another study to look at that indicates that a CBD-rich extract of cannabis actually had a greater effect on treating epilepsy than did purified CBD, another study supporting the concept of the entourage effect. So in essence, this was a meta-study. They looked at a lot of other studies, and they found that there were more reports of improvements in epileptic patients that were treated with complete plant extracts as opposed to patients treated with purified CBD. And patients with CBD-rich extracts, as opposed to pure CBD, actually required a much lower dosage per day in terms of the amount of cannabinoids they needed in order to find efficacious results. And furthermore, adverse effects were more frequent in products containing purified CBD rather than in whole plant extracts. So this study, again, says that there are some synergistic effects between CBD and other phytochemicals in what they term here the entourage effect. Here's another study looking at anti-tumor action of pure cannabinoid, so a purified CBD extract versus a botanical drug preparation in models of breast cancer. They find that cannabinoids, they state that cannabinoids trigger anti-tumor responses. That's kind of well-known background prior to this study. But they found that the botanical drug preparation, as they call it, the whole plant extract, was more potent than pure THC in producing anti-tumor responses. And their results suggest that standardized cannabis drug preparations, so whole plant extracts, rather than pure cannabinoids, could be considered therapeutic to manage breast cancer. Terpenoids, turning and looking at a slightly different aspect of what we could umbrella under the entourage effect, is the effect of terpenes and terpenoids. So cannabinoids are well known to have anti-inflammatory effects, but it turns out that some of the terpenes and terpenoids found within cannabis are pretty well known to exert anti-inflammatory and anti-nociceptive activities, both in vitro and in vivo, so in lab and in actual animals. Although none of the essential oils they tested in this, so none of the terpenoids that they tested in this study worked as well as a pure CBD extract did alone. Flavonoids are another interesting and largely understudied class of phytochemicals produced by the cannabis plant. And as of yet, there's not a ton of research done on on flavonoids, but some of them are known to have antioxidative effects and anti-inflammatory properties. Cannaflavin A and B in particular are known to exert anti-inflammatory activities. They're actually known to be 30 times more effective than aspirin in some regards. So these cannaflavins actually may contribute greatly to the overall effect that you get from a whole plant extract as opposed to purified cannabinoids. It's definitely an area that needs a lot more study. But to summarize, I really like to take the title of this Dr. Russo paper. Cannabis is more than simply delta-9 tetrahydrocannabinol. I think that's the best way to sum up what the entourage effect truly is and what the studies have shown to be the case. So flavonoids, again, as Dr. Russo says here, have strong anti-anxiety effects without a sedative effect. And he says here that after talking to estimated number of 200 patients who have used both Marinol, that synthetic pure THC molecule preparation that's available pharmaceutically, that cannabis, as compared to Marinol had nowhere near the efficacy of pure cannabis, according to these informal interviews that this prolific author has conducted. So in conclusion, it's been well established at this point in the scientific literature that there is, in fact, a difference in terms of the biological activity and the physiological effects of purified cannabinoids as opposed to full plant extracts. And whether this is due to different molecules potentiating the effects of binding of THC or CBD at the receptors at which they bind, or whether it's due to some of these chemicals binding to other receptors or all sorts of different mechanisms that are as yet undescribed, it's clear that regardless of the mechanism, something is going on, and purified THC or CBD doesn't seem to have nearly the same therapeutic effects, and by all accounts, it doesn't give anywhere near the same subjective experience to the user as does a full plant extract or simply smoking cannabis. Cannabis is more than simply delta-9 THC. And this is an area that I really hope we see lots more research on in the future. And it really ties into this idea that I keep bringing up in our discussions of designer cannabis strains. I really feel like the medicinal potential of cannabis lies in the future, understanding what these different phytochemicals do and how we can engineer a cannabis strain to have a particular blend of phytochemicals to have a particular end result to treat particular medical conditions. So, in about 15 minutes, that was the Entourage Effect. Thank you for watching.